Hi all, I am Sasidhu Alakon from the Department of Computer Science and Engineering, University of Morocco. My intention today is to present to you the paper we have compiled on enhancing plasmid chromosome classification with graph neural networks. It is titled as MetaGraph. First, I am going to give you with background information relevant to the research field. For centuries, lab culturing has been the primary method of extracting bacterial samples necessary for microbiological experiment. In that procedure, we implant small bacterial communities in favorable environmental conditions such as nutritious gel mediums. After the communities grow sufficiently, they are used for experiments. The main issue with this method is that only 2% of all identified bacteria are culturable. Metagenomics facilitates the analysis of microbial samples collected directly from the environment. It allows scientists to explore more into the microorganisms. But unlike in lab culturing, due to the diversity of bacteria, the extracted DNA in metagenomics are bound to be more like a mixture of DNA. There are two main sources of genetic information inside a bacteria cell, plasmids and the chromosome. There are considerable differences between plasmids and chromosomes both structurally and functionally. Length and circularity are some dissimilarities regarding the anatomy. On the functional side, chromosomes mostly pass information from a generation to generation, while plasmids are capable of sharing genes among the same generation. The metagenomic procedure of retrieving genomic information from samples contains several steps. DNA extraction. The initial step of this process has improved heavily with the introduction of next-generation sequencing techniques. These short-length reads are assembled using assembly graphs into contigs that are comparatively much longer. Let's look at some literature related to the problem. Plasforest is a plasmid chromosome classifier which implements a machine learning based approach in order to acquire better results. It trains 3 different artificial neural networks using 5mer, 6mer and 7mer counts as features for each model. A combination of a prediction is taken to get the final output class. Plusclass is another tool which classifies plasmid and chromosome contigs based on logistic regression models. It uses 3 different logistic regression models to get the plasmid chromosome prediction for sequences of different lengths. GraphPlast try to improve the trade-off between precision and recall often seen in other plasmid chromosome prediction tools. They generate an assembly graph using the DNA sequences. The high confidence nodes are labeled using an existing tool like PlusFlow or Plusclass. They use a k-nearest neighbor classifier to classify the low confidence nodes into plasmids and chromosomes. This is a simple summary of the literature that we looked at. While PlusFlow and Plusclass use sophisticated machine learning models, their approach still has room to improve upon using further enhancement techniques. GraphPlast tries to achieve that by using k-nearest neighbor algorithm on the adjacency assembly graphs generated. Now we are moving into the methodology of MetaGraph implementation. MetaGraph enhances plasmid chromosome classification using graph neural networks. Following diagram shows the summarized view. MetaGraph takes the DNA sequences and initial predictions as the input and output the enhanced plasmid chromosome probability. Before going to further details, let's discuss few related terminology. What is a graph neural network? Graph neural networks are a type of a neural network designed to operate on graph data structures. Graph neural networks can be used for graph level, node level and edge level predictions. MetaGraph uses node classification as the graph neural network technique. In node classification, every vertex in the graph is associated with a label. This label is used to get the correct classification class. Then the graph neural network will train according to these labels. During the training process, each graph node will aggregate the feature information from its neighbor nodes and update its own features. Since the model consists of multiple layers, This process happens multiple times where the value of a node is propagated amongst the neighboring nodes in each layer. An assembly graph is a bidirectional graphical visualization of the metagenomic read assembling process, which is based on the deep green graphs. The nodes of the graph are the DNA sequences, and the edges are created using the overlaps between the DNA sequences. Following diagram shows a high-level view of the MetaGraph. MetaGraph takes the DNA sequence files and initial prediction as the input. DNA sequence files contain the following information regarding the DNA sequences: complete sequences in FASTA format, detail of overlap between sequences fragments in GFA format, mapping of sequences with their overlapping fragments, which is used to generate the assembly graphs. Initial prediction contains the plasmid probability for each DNA sequence generated by another plasmid chromosome detection tool. It contains both low accuracy predictions and high accuracy predictions. The first step of the MetaGraph procedure is to generate the initial assembly graph. In the assembly graph, each node is represented a DNA sequence. Then system reads the input GFA file and identifies the DNA sequences with overlaps, and edges are added between DNA sequences with overlaps. The redundant edges and self edges are ignored in the process. The next step is to calculate composition-based features for each assembly graph node. MetaGraph used former counts as the composition-based feature. Then the next step is to label the graph using initial prediction input. The main goal of this step is to identify the sequences with higher confidence for the model training step. A node will be labeled as Plasmid if the initial prediction probability for that node is higher than 0.8. A node will label as Chromosome if the initial prediction probability for that node is lower than 0.2. Other nodes will be labeled as unclassified. The label represents the class of the DNA sequence. The graph neural network is trained using plasmid and chromosome vertices. Then the trained graph neural network is used to classify the unclassified vertices. MetaGraph uses node classification as the graph neural network technique. Each graph vertex has plasmid probability and normalized former frequencies as features. These features are used to train the graph neural network. MetaGraph uses 4 layers in the graph neural network model. There are 3 graph convolution layers and 1 linear layer. The system trains the model using the training dataset in real time. We use 20 epochs for the training process. We use cross entropy as the loss function for the training process and Adam as the optimizer for the graph neural network. The final output of the MetaGraph is the enhanced plasmid chromosome probabilities for DNA sequences and classify DNA sequences into plasmid or chromosome based on it. Following table shows the datasets used by MetaGraph. There are both simulated datasets and real datasets. The real dataset used is based on wastewater plasmidome. Now we are going to discuss about the results of the MetaGraph. We evaluated MetaGraph on the set of simulated datasets and the real dataset introduced in the previous section. This table compares the results by using only plus class versus enhancing plus class results with MetaGraph. MetaGraph has been able to improve upon both precision and recall of the plus class results, hence resulting in a higher F1 score. When the graph is highly connected, the improvements to the results are high. This is due to the fact that the graph neural network can propagate the features better when its connectivity is high. This diagram shows the percentage improvement for 2C5P dataset. The result propagation for the 2C5P dataset using assembly graphs shown in this diagram. The red color nodes are the chromosome predictions, while green color nodes are plasmid predictions. The white nodes denote the low-confident unclassified nodes. Figure A shows the prediction generated from class. The graph obtained after applying threshold of 0.2 and 0.8 are shown in the figure B. The final predictions obtained by training the graph neural network are shown in the figure C. The ground truth values are shown in figure D.
Now on to the conclusion of our research. As the result of our research, we derived that GNNs can be used to improve classification of plasmids and chromosomes. And that's the end of our presentation. Thank you all for the opportunity to present our research in this conference.